Good morning to everyone. I'm Luca, and today we are going to discuss the paper Local Slow Waves in Superficial Layer of Primary Cortical Areas During REM Sleep, published by Funk et al. In 2016. I'm going to start with the introduction, then Claudia will take care of the methods, and after that, Rosanna will discuss the results. In the end, I will come back for the conclusion. Before starting with the main content of the paper, I think that it's useful to refresh some information about the sleep stages. The figure on the right reports EEG recordings during a night of sleep. As you can see from the point of view of neural activity, sleep is not homogeneous throughout the night, but it's made up of cycles of 19 or 120 minutes each, composed of different phases. As soon as you close your eyes, brain waves begin to slow down and you enter the light sleep phase, where eye movements stop, temperature drops, and there are bursts of electrical activity, as you can see here in the N1 phase. As you continue to sleep, you enter the deeper stages of sleep, N2 and N3. And as you can see from the EEG, brain activity has low frequency and high amplitude in N2 and N3 stages. The stages from N1 to N3 are called non-REM sleep, which is characterized by, from a neural activity point of view, slow wave activity, which is generated through recurrent connections within the cerebral cortex, where cortical pyramidal cells excite one another in a positive feedback loop. This recurrent excitation is balanced by inhibition, resulting in the active state of the slow oscillation of slow wave sleep. Then we have sleep spindles that you can see here in the image in the N2 phase, which are bursts of 11 to 15 Hz activity in the EEG. They are characteristic transient feature of N2 sleep and are believed to mediate many sleep-related functions, from memory consolidation to cortical development. Finally, we have K-complexes, which are long delta waves that last for approximately 1 second and have a function in maintaining sleep and memory consolidation. From a behavioral point of view, NREM sleep is characterized by relaxed muscles and sensory disconnection, which means that the person who is sleeping is less responsive to stimuli and is more difficult to wake him up. After the non-REM phase of sleep, approximately 90 minutes after sleep onset, it comes the REM phase, which is very different from the previous one. First of all, because it presents a wake-like EEG. As you can see here in the picture, REM sleep and wake has very similar EEG recording. And furthermore, during REM sleep, eye movement increases. Arms and leg muscles are paralyzed, but our brain is active, producing vivid dreams. Finally, even during REM phase, there remains a strong sensory disconnection. And the presence of sensory disconnection has always been considered a paradox, because low waves, which favor sensory disconnection, are not present in REM sleep. So as I was saying, the absence of slow waves in REM sleep and the presence of wake-like activity suggests that all cortical neurons are tonically active and cortico-cortical connectivity is preserved, which means that there should be no sensory disconnection. In addition to that, cortical activity during REM sleep was thought to be homogeneously wake-like. The aim of this paper is to show how, using local laminar recording in freely moving mice, they discovered that slow waves occur regularly also during REM sleep, but only in primary sensory and motor areas, and mostly in layer 4 and layer 3. So this could mean that the focal occurrence of REM slow waves activity, specifically in the layers of primary sensory cortex involved in feed-forward signaling, may explain both sensory disconnection and dreaming. So this was the introduction part, and I gave you some background information, and now I will leave my place to Claudia. We will talk you about the methods.
Hi everyone, I'm Claudia, and I will guide through the methods of this study. The study involved recording brain activity in mice that were moving freely. Mice were not restricted to avoid bias while measuring muscle activity. These mice had EEG electrodes in frontal and parietal cortex, EMG electrodes in neck and vibrissae muscle, and laminar probes in various cortical areas. The goal in this first part of the experiment was to examine general patterns of neuronal activity during the sleep-wake cycle and following sleep deprivation. Sleep was scored in 4-second epochs with a focus on consolidated sleep episodes during the light phase, when mice sleep most frequently. In the figure, we can see a 24-hour hypnogram showing the distribution of consolidated sleep and wake episodes in one representative animal. All consolidated non-REM and REM episodes during the light cycle were used for analysis. In the second part of the study, the goal was investigating specific patterns in the regular sleep-wake cycle and during period of sleep deprivations. To survey regional cortical activities across the behavioral state, they examined local field potential, that are electrical signals measured in the brain by placing electrodes within or near specific brain region. The local field potential were recorded from layer 3, the external pyramid layer, of 6 cortical regions using laminar probes. The technique of laminar probes, including electrode designed for acute extracellular neural recordings, in which the shaft of the probe can be easily implanted in several brain areas of rodents or higher mammals without causing serious brain damage. The multi-electrodes provides high-quality electrophysiological measurement, including local field potential, multi-unit activity, and single-unit activity recordings. The multi-unit activity is a measure used in neuroscience to study a collective electrical activity of a group of neurons in a specific brain region. As I was saying, they recorded from 6 cortical regions, including primary visual, sensory, and motor areas, secondary visual and motor areas, and retroscranial cortex, an association area involved in memory and spatial navigation. They choose to focus on REM waves because such activity may not have been previously recorded, since unit intracortical recordings are usually biased toward deep layers, where the typical wake-like state can be observed. At the same time, simple EEG tackles very superficial layers, 1 and 2, where such slow waves cannot be observed either. It is also to be noted that the restricted area in which slow waves can be observed during REM may explain why they have not been observed before. Currently, technology can only measure one layer at a time, often on the surface or deep in the cortex. But as we can see in Figure 8, new electrodes are in the works to measure different layers simultaneously. Here we can see extracellular recordings made from the primary somatosensory cortex of freely behaving rats using a flat microelectrode array with 16 channels that cover all the layers through the cortex. The signals were preamplified through a digital edge stage and filtered. Exemplar extracellular spikes were identified through spike sorting with principal component analysis and k-means clustering. The shaded area around the sorted spike waveforms indicates the standard error of the mean. Now I will let Rosanna continue with the results.
EEG and EMG showed global patterns just like described during our introduction. Laminar probes, however, paint a different picture. In Figure 6, you can see how wake presents characteristic low-voltage fast activity in both surface and deeper layers of the brain, the same that we can also find in deeper layers of the brain during REM sleep. During non-REM sleep, however, we can also find the characteristic spindles as well as the slow waves between 0.5 and 4 Hz, the same type of wave that can be also seen in surface layers during REM sleep. Couple of differences: these slow waves tend to be shorter, and also they do not seem to present the characteristic spindles. This is very significant because it puts forward that patterns in the brain during REM sleep are not as global as previously thought. In fact, surface layer and deeper layers behave rather differently. To characterize this discovery, the researchers have collected the local field potential data, and they conducted a spectral analysis. Spectral analysis is done to examine different frequencies that can be present in electrical signals generated by groups of neurons in the relevant sensory areas. Spectral analysis allows the researchers to have a better understanding of the cognitive process in the brain, as they are associated to specific frequencies. It is also useful to identify patterns, as we will see in a moment. If you look at the graphs, on the left side, you will see the global patterns data, and on the right, you have specific brain areas, secondary and primary areas alike. But in both, non-REM sleep is represented in black, and REM sleep is represented in red. On the horizontal axis, we have the frequency, and on the vertical axis, we have the relative band power. It represents the strength of the electrical signals within a specific frequency range, and it is measured in microvolts squared. Hence, you can see the negative powers of 10. In other words, the graphs represent how the power of the signal is distributed over the frequency. In image C, you can see how brain activity is progressively decreasing during non-REM sleep, while it peaks a couple of times during REM sleep, just as expected. In image E, on the other hand, we can clearly see how V1, S1, and M1 show different patterns during REM. Not only the peaks are not as marked as during the global tendency phase, but also they seem to be closer to non-REM patterns. The difference is even more striking if we take a look to the secondary layers, where the difference is far more marked. So to further characterize the behavior of such slow waves, researchers have tried to investigate whether they were coordinated along different areas or layers of the brain. It was found, however, that slow waves don't seem to be coordinated across brain areas and layers. Quite interestingly, though, they found out that slow wave activity during REM sleep seemed to remain unchanged despite varying wake duration and sleep pressure. In figure 7, you can see at the very top 2 spectrograms representing slow wave activity during a regular wake-sleep schedule. On the x-axis, you will find the time expressed in hours. So at the bottom, you will see all the hours the rats were sleep deprived. On the y-axis, you see the depth at which the electrodes were placed, and it is expressed in micrometers. Brighter colors indicate more activity, a higher amplitude of activity. So right here, you can clearly see how the brighter color represents that at the very beginning of sleep onset, slow wave activity is higher because bodies are tired. And this tiredness decreases as we keep sleeping, and therefore also the colors fade away. During REM sleep, we can also find a difference in color, but not quite a clear decrease pattern. This becomes even clearer these non-REM and REM slow waves are when we take a look at the very bottom. We can see after sleep deprivation, non-REM slow wave activity is definitely more intense at the beginning, meaning our body is more tired and therefore the slow wave activity is higher, and the same type of decrease patterns can be found. During REM, however, not only the intensity has not changed, but also the decrease pattern is completely off. There is no decrease pattern here. And before we go ahead and try to explain the reason for this, we have also to introduce another interesting and relevant data, which is about the layers. Now, the researchers have later noticed that in layer 4 is where the slow wave activity peaks. Not only that, slow wave activity seems to progressively diminish as we go further from layer 4. And so researchers have posited that these two things, so slow wave activity being sleep pressure and the peaking in layer 4 may be connected. In fact, I think that the homeostatic regulation of non-REM slow wave activity arises when synaptic connections between brain cells strengthen during prolonged wakefulness. So, since in layer 4, the synapses may not be as plastic throughout all of our life, so they may have the tendency to be rigid during adulthood, they may also be less responsive to the changes that take place during wake. Therefore, this may be able to explain why REM slow waves are insensitive to sleep pressure. I pass the mic to Luca for the conclusions.
In conclusion, as we have said in the intro, REM slow waves could explain the paradoxical sensory disconnection we experience during REM sleep despite a wake-like EEG. Previous studies have already revealed a strong connection between increased level of slow wave activity during REM sleep and an increased effectiveness in disconnecting from external sensory input. These findings are sustained by multiple studies involving rats, where researchers observed that arousal thresholds during deep REM sleep were notably higher compared to the REM sleep, with specific mention of their response to auditory stimuli. On the conjecture that a similar function could be performed by slow waves during REM sleep should be able to explain also why primary sensory areas are specifically impacted. In the picture, you can see how a separate experiment accounts for possible different populations of neurons being involved in the feed-forward and feedback process. Considering that secondary association areas are still active during REM, this one-sided inhibition may bias the balance between bottom-up and top-down cortical signaling in favor of top-down, potentially accounting for both sensory disconnection and vivid dreaming. So this was our presentation. I want to thank you for your attention, and we hope that we will have a good conversation and a good discussion. Thank you again.